Um, are we doing lichens tonight?
Lichens and member fines.
Okay, so I sent these lichens back in February, and you weren't around. I don't think we ended up looking at them, but we might have, but I don't think so. I have very little to say about them, so I'm hoping maybe you'll have more. And if I did show them already, I apologize. So these were in the Chiricahuas in southeast Arizona in February. And I have only to say the name that INAT has provided. And so, if we are talking about lichens, I've got lichens, but I need people to help talk about them.
Yeah, so I don't know Arizona lichens, but I can definitely see several different species here. There's the big brown foliose lichen in the center of the photo. There's the green, green, yellow, kind of finer folio stuff around the edge. I see an areolet uh, crustose lichen over here on the right.
Is this on
Um,
rock?
Yes. And
so if we were in New Jersey, the first thing I would think of for this one in the middle would be something in the genus Lysallia, but I wouldn't. I don't know what else there is out in Arizona that we don't get that it might be.
what did I not say much
You just
or
have it to family
okay. Yeah. Um,
for this
areolate
guy.
is the one on the far right.
Yeah, so it's kind of cracked into these
Ah, correct. There. Yes.
little distinct polygons,
Mm-hmm.
kind of like scales almost.
Cool.
Yeah.
Yeah. And the black one has like little bumps on it or
Yeah, like warts.
yeah.
Yeah, so that's we get two species in the genus Lysalia that has those warts that are folios that grow on rock up in northern New Jersey. So that's the closest thing I can think of, but again, I don't know
Okay.
what other genera are out west that I don't know.
Okay.
And
This guy.
do you have any guess on the areolet one? It looks like it had some black uh apothecia, right?
Yeah, there's a lot of those. Even if we were in New Jersey, there'd be, you really got to look at the spores for those.
Ah, cool.
And there's the one with the white thallus and there's the one with the gray thallus. Those might be different. So you probably have at least four lichens in this photo. You've got this yellow green one here that's got kind of broader lobes, and then you've got this one over here that's kind of finer. Those might be two different species as well.
Mm.
You
And then
see where
that
like this looks like the finer one here, and then everything next to it looks like the broader lobed one.
what kind of looks like dusty blue green between the areolet one and the folios deals? Yeah, right there. Is that maybe just the green folios one? A different like
This right here,
yeah.
no, that's definitely Crestos
Oh, okay.
right
I
here where
thought
my cursor
I thought
is.
thought it maybe was like the Ceridia of the green one.
No, I don't think so.
Cool.
Yeah, and you can see that dark line that separates the two Crestos lichens here. That's
Ooh.
where they're
Is that
Let's
dark
see.
line like I don't know, for lack of a better, is that like a spalting?
It's my understanding that there's a name for it. I can't remember the name, but my understanding is that they're basically competing for space here and they're exuding chemicals to fight for space basically. So it's basically like a chemical warfare front between
Is
these
it
two lichens.
is it a melanin like mushrooms?
I don't know. I don't
I
know
mean,
what
it
the
might be.
substance
It's that
is.
dark color.
Yeah. But you can see this between individuals of the same species or between individuals of different species. It's basically just the point at which they're competing for space. And usually only see it between two crustose species.
Hmm.
I've have read that it's probably a melanin compound.
Yeah, makes sense.
Nice. So it is a similar thing happening.
Cool.
Okay.
Next up, we got this guy.
Yeah, and I think the genus may be right. And then there's the black on the left.
Mm-hmm. Yeah, I'm not familiar with. We don't get this genus in New Jersey, I don't think.
Do you get any cobblestone lichens?
Um, I don't know. I'm not familiar with that common name. I only pay attention to the genus names. We definitely don't get any Pleopsidiums. I
Okay.
don't know if cobblestone refers to any other genera
Mm-hmm. Yeah, I
so
mean,
far.
it might. It's the look, I think it's referring to, but
So, Jason, why is this genus not in New Jersey
I mean, you can see there's something here.
or the East Coast? Seems like very sparse.
Yeah, I think this is probably a misid. Yeah, that's definitely not. So, um, yeah, I think some of these that are further in the East Coast are probably misidentifications. I don't know why do some, I mean, you see that distribution with a lot of species and why they're, on the West Coast versus East Coast. Um,
yeah, Arizona has a lot of lichens. That's my understanding. I read up about it when I got back, but and so I didn't again, but I do remember reading that there's quite a lot of lichen diversity there.
yeah. Yeah, it's not just diversity, but the uniqueness of the species that are there.
It's
Um,
surprising. You wouldn't think so.
yeah. Yeah, I don't know. I mean, we do get, examples of lichens that span the whole continent, east to west. But yeah, what's the limiting factor on the eastern boundary? I'm not sure.
Oh,
Right.
oh,
We
this
got a
is
mushroom.
old. Yeah, I didn't. Yeah, um, maybe we did look at these. These were growing in my shiitake bed, and they are again. They're out there right now as well.
Hmm.
Um, I'm pretty sure that they're the same thing, but I think it's Xanthoparmelia. But
Okay.
I definitely have more lichens. Oh gosh, we shouldn't look at this. What else do we have?
All
Here,
right, here.
here's another lichen. Oh, this one's cool. Yeah, next to the other one.
Yeah, so we do get Candelariella in New Jersey. Um, but this doesn't look like any of the species that I know.
Mm.
And
So those are little orange apothecia. Yeah,
yeah,
that's cool.
yeah, I don't know. I can't even confirm the genus on this guy. Again, the lichens of Arizona are so different than the East Coast.
Do we get any of those? Because I always see photos online of some of these foliose ones that have the bright orange or kind of yellow, yellowish apothecia. Do we get those in New Jersey on like? Because is this crustose or foliose? Are there
Yeah,
little
that's a good
like
question.
they're little
It's not embedded
squamules?
in the land in the substrate, so it doesn't look crustose to me. I mean, it looks like I could get my knife under there and peel it off, right?
Yeah, it looks like there's little lobes at the margin, right?
Yeah. So I'd call it foliose. Um,
But yeah, do we get any of those brightly colored foliose ones in New Jersey with like the yellowish or orangish apothecia?
Yellowish apothecia. I mean, we definitely get yellow-orange lichens that are ceridiate or asexual. Let's see, does
You showed that one that you thought was a misidentified cobblestone that was yellow from, I think.
right, so that could have been a Xanthomendoza species.
Yeah,
So like
which
these,
I would guess Xantho Mendoza like they have you know they reproduce sexually, so they're gonna have some apothecia, but like that thing that we just looked at that Hunter showed, it was like
it's
tons of
got, yeah, 90% coverage. Um, and most of the Xanthomendozas typically grow on bark. Let's see, Ari, how do you spell it? Yeah, Candelariella, like these two, efflorescens and vitulina. But even so, I mean, it's not that dense apothecia covering. Usually this grows on bark, not on rock like the other one. So I would be surprised if this is a Candelariella, but sign in.
Okay. Yeah. There's
Let's just
definitely a lot of
see.
good information out there on Arizona lichens. I just noticed today it was on lichens. I didn't realize. And we didn't really ever look at these because we were hoping you would be there. And so I'm
Yeah,
sorry.
this looks like a better match.
Yeah.
Squamelia subsoluta.
Oh,
And that one's
that
not
does
bad
too.
either. Yeah, actually, this one looks like a closer match.
Mm-hmm. Well, can we see where they're showing that? It looks like Arizona, maybe.
Yeah,
Yeah.
yeah,
Cool.
yeah. I'll throw that on there. You can take a look at it just so you have it.
Thanks.
Yeah. And then let's see, and then this guy. So, oh man, we got a ton of them in here.
Yeah, and this black one, do you think it's different than the first one we looked at?
I do.
And is that foliose?
Yes.
Okay. And then what's like fructose? That's one too, right?
Fruticose. Yeah. So those are going to be
Fructose.
the three-dimensional branching or drooping ones.
Oh, okay. Mm-hmm.
And I'd be surprised, a lot of when you're out in the desert, I'd expect a lot of the lichens to be tightly oppressed to the substrate. So it wouldn't surprise me to have very few fruticose lichens out there.
Got it. Makes sense.
But yeah, I see one, two, three, four. Let me take a closer look at this stuff here to see if that's a fifth one. But yeah.
So I mean, the light green one looks the same as what we looked at before, but the orange is different than
Yeah,
than the bright
because that
orange.
doesn't have all the apothecia on it,
Mm-hmm.
but it still looks foliose. Could
And
just
it
be
looks like, yeah,
it could
different
be
than,
infertile, but I'd be surprised if it was this substantial and it didn't have apothecia like the other one. If it was the same thing. So
mm,
yeah, probably something different.
okay. Well, I'll try to learn more about Arizona lichens if I keep going there.
Yeah. This is
I like in the dead center of this photo, you can see the orange one growing on the directly on one of the black ones. Is that what we're looking at there?
Yeah, look at that. Yeah, at first I was gonna say, no, it's the black one on the orange, but you're right.
It's pretty cool.
Yeah,
yeah.
that's different. Hmm.
Yeah, so the let's see.
Yeah, and this rock is, I mean, it's a very exposed. You would think it was like maybe protected and a cool, damp environment or something, but it's like, no, it's sun baked every day and um, very exposed to the elements.
Yeah. So one really good resource for lichens of Arizona is the consortium of the lichen herbaria, where if you search for taxon, you can search for a lot of them, but um, all these descriptions come out of the 2004 lichen flora of the Greater Sonoran Desert region. So you can get a lot of really good information from this website if you don't have this 2004 publication.
Okay. Um, but you kind of have to start with a name. I mean, basically,
On the website, I think so. I've never tried. Um, yeah, I don't
or
think it
maybe
has
a
keys. I mean, the I don't know if this document has keys. I mean, usually if you have descriptions, you got keys, but I've never seen keys on this website. I've never really looked either because I've never studied the Sonoran lichens.
right. I think that document might, because I think I did come across it originally.
All three volumes made available here for download as PDFs.
Cool. Oh, good. There's a key.
Yeah, yeah, the genre. It's a big a place to start.
Yeah, thank you.
Yeah,
Yeah, I should do that. I should be going there another couple of years at least in February, I think. So,
cool.
yeah, so hopefully next time I'll know a little more.
Right, thanks, Hunter.
Yeah, thank you.
Let's see, we've got some for Dave, but I don't think he's going to be joining us this evening. So maybe we'll skip his and come back to him at the end. Dorothy, is this one of yours? You want to go next?
Well, I guess it's a jelly skin. I'm going to be asking you what I photographed. This was very wrinkly. So is it the old genus Leptogeum lichenoides or not?
Where was this taken?
Jenny Jump.
Okay. So from Jenny Jump on rock.
Yeah, these were on rock.
So let me look up.
I think you have it listed as Scitinium or Scitinium.
No, so this one I believe is the one that is likely Colema
Oh,
subflacidum.
all right. So my other picture shows something flatter. And when we looked at that rock, that's the flat stuff.
Yeah.
I thought you said there were two different ones. So I don't know these black lichens at all. I don't see them ever.
Yeah, I think this might also be Colema subflacidum.
So it can look wrinkly, but it can also look flat like that.
I think so. The Scitinium is this thing here. Scitinium lichenoides.
Oh,
So it's
just
kind
that
of like
one.
mixed,
Okay.
there's some here. It's kind of like mixed into the moss. So that's the Scitinium lichenoides. So if we pull
Okay,
up
that's a different picture. Right?
yeah, so different than this.
And then there was another one that I sent in a separate.
I saw 3.
So that's the Kalema 2.
I think so. I could be wrong, but
That was on another rock.
I mean, usually when I see Colema subflacidum, it's this wrinkly one.
Oh,
So
okay.
this guy is kind of the oddball out. If there's a third lichen here, it'd be this one. I think this is the Colema subflacidum or something similar. I don't know how you tell the subflacidum apart from the other Colema species, but I think subflacidum is the species that this is most likely to be. And then this guy is definitely the Scitinium lichenoides.
Okay, just up
Um,
in the top there.
but let's
I
pull up
I have trouble with my cell phone. It only takes clearer pictures on portrait, and so I can't zoom in very well.
it's not a great shot. Let's see if we can get a nice shot of what this thing looks like. Yeah, so there's the
Oh, is that
Scitinium
yours or
lichenoides.
Dave's?
No, this is John Franklin's from
Oh,
up in the Catskills.
so this is it's very lacerated, or
Yeah, it's almost like ciliate. I guess that's Isidia. But yeah, it's usually like this really coarse, rough
okay,
stuff
and then
intermixed with mosses.
and when it's wet, it's going to be gelatinous.
Yeah, kind of like this. It'll be kind of I don't know about gelatinous, but it definitely gets kind of moist looking.
Okay,
Or the apothecia. But yeah, it's difficult to even recognize as a lichen if you don't know what to look for.
Yeah, it looks like dead
Oh,
moss.
Yeah.
That's what I was just gonna say. I've probably seen this, but it just looks like dead moss.
right,
Mhm.
right. You
Yeah,
mentioned
usually I usually see it on limestone rock. Um, and you can look at it closely and look for the isidia or apothecia.
Yeah, you said it at the time that it looks like the dead moss.
Yeah.
So is that like from that last picture? I mean, it's a foliose.
Yeah, it's foliose.
Yeah. These are some good pictures. Yeah, close.
Yeah. Yeah, it's not super common. I would only expect it to be up in northern New Jersey, and I usually only see it in limestone bedrock. It's a good picture.
Okay, thank
Cool.
you.
Yeah, thanks, Dorothy. All right, anybody else have anything to share?
I can
We're
share
good.
some other stuff. If no one else has stuff to share.
Sure.
Oh,
From your screen? Want me to
um,
stop sharing?
we could try that.
Or you could send it into the Taxonomy Tuesday email.
Okay, yeah. If somebody could could we look at one of Dave's or something while I do that?
Yeah,
Okay,
yeah, we can
okay,
Yeah,
I'll
I'll
get
I'll
on it.
see what.
I'll put something in the chat if we want to look at it real quick. It's not a lichen,
Sure.
it's just an update on my slime molds.
Yeah,
Cool.
let's do it.
Everyone, that's in
Jason,
the
do you
chat.
have any pictures of lichens that you took at Jenny Jump or not?
No, I didn't really take a lot of pictures because I was too busy talking and answering questions.
Oh, okay.
All right, so I showed this last week, um, and you're gonna have to zoom in. Uh, they're just starting to open up. They're very small, but I think you can make out in the ones in the center that they're revealing a little bit of a black mass in the middle there. I called it Diderma because Diderma is supposed to have, well, what, two skins, right? It's got two layers of Pyrrhulum. Um, but I actually think this is just kind of an immature Physarum cinerea, um, just based on their clustered and sessile. But yeah, this is from moist chamber culture I showed last week.
Cool.
Yeah, just starting to crack open. Uh, Hopefully, I can get these to sporulate and actually reach maturity, but they're not quite there yet. But
I did go as far as collecting like one like Kagala or like Ogala or whatever, um, to do some microscopy on because I was inspired by your
good,
yeah.
yeah, awesome, yeah. Slime molds are cool, and these moist chamber cultures, like I said last week, they're really, really fun. I have close to 20 cultures that have healthy plasmodium in them. Yeah, and this shot you can see that they don't have stipes; they're sessile, right? They're just growing right up against the Tupperware that I'm using as a moist chamber culture.
It's funny they prefer that to the other substrate.
Yeah, the other funny thing about this particular species, because I did send this to Dr. Stevenson, and he said based on their sessile, they're closely clustered, that's what he thinks they are, although, they need a bit of time to actually show themselves. Um, that, and I remember it from the class, prefers live like herbaceous material. So like it likes the stems of living plants. Um, so if you ever find a little tiny gray slime mold on a living leaf of like a plant, it's a good place to start. And then there's the plasmodium there. But yeah, Hunter, like you were saying, it's weird that it prefers the plastic when it's something that in nature prefers living herbaceous material.
The plasmodium is where, Brandon, that white thing?
So it's the white thing, and then there's also yellow plasmodium between the two leaves. Just
Oh, I see it. Mm-hmm.
yeah,
Oh,
yeah. Try to get a 2-for-1 in there. So there's three actual plasmodiums in this photograph. If you zoom out, it takes a bit of imagination to see the brown uh, streaky ones on the paper. And maybe you can't make them out in this photo, but yeah, there's also brown plasmodium that really likes the underside of the paper in this culture.
Have you ever seen those fruiting bodies on anything else?
No,
Like,
maybe once on where it should be in nature on like the leaves of a uh, in the garden of my apartment place on living like tulip leaves.
the whole thing's a bit befuddling. Honestly, I love it.
Mhm.
Uh,
Yeah,
yeah, I'm like, what? Like, where else would you have seen these?
but
Because
that's
like
but that's a good
you have collected this thing, like not knowing what you have. You're just fishing,
just fishing. And
which is crazy town.
and honestly, I was shocked after Steve. That's what he said. Call me Steve. Steve um, gave us a couple pointers, and just those couple pointers, he was like, "Yeah, I have like a 90% success rate," and that's about where I'm at this point too, which is
good.
Put stuff in box, stuff in box. It's wild.
So Brandon, how important is the plant species in the culture? Like these look like red maple leaves. Are you going to get a different suite of slime molds if they were oaks or sweet gum?
Oh no, he just left.
Oh, yeah, looks like we lost him.
Anyway, Brandon's got to head up some kind of club pet keeping thing. I love this. I think this is hysterical.
I do too. I'm so intrigued. I do want to try it.
I mean, maybe I could get away with it in the garage, but that's about it.
Right. Yeah.
All right, let's see.
Yeah, and that was a good question. We sort of talked about it last week too. Uh, I mean, yeah, it's just you don't know what you're gonna get at this point.
Mhm.
But he did say that you can kind of predict based on the substrate what you might get, but he doesn't know that yet. I think he said that last week. But at this point, he's just fishing, right?
yeah. I'm back. I lost you guys for a second. Sorry.
Yeah. Yeah, I was welcome back, Brandon. I was just asking how important the plant species was. It looks like these are red maple leaves.
Yeah, and this is from submerged material, which is that was one of the tricks you pick up is what do you culture? Because I was pretty much just culturing things where I had already seen slime molds. Um, and yeah, one of the pieces of advice was collect material from streams, sticks and leaves from streams or ponds and things like that. And this one, uh, it's got three different types of plasmodium in it, and some fungi, which is always fun.
Hmm.
So I mean, oh man, are they aquatic-ish?
They just persist, I think. If some plasmodium is on a leaf and falls into the whatever, stream or pond or whatever it may be, um, those spores are still, or that organism is still alive, and it can go into this kind of hibernation mode, and that plasmodium just kind of shuts itself down, and then all of a sudden, it gets in the right conditions that it wants the fruit, and yeah, it's just always kind of on there, I think. Or maybe there was a rogue spore uh, from each one of these species.
So all you're really doing is creating the right conditions, and it's just so very easy to do.
Yes, it. Yes, and if anyone watches that recording from last week, the important thing that I never did before uh, I learned a couple of these tricks was you need to flood it overnight. So almost submerge the material in water overnight, then drain it, and then make sure that it stays out of the sunlight. You want to keep it airtight as much as possible. Um, but, you know, I think opening it up every now and then is good to promote fruiting and sporulation.
And you said that so it's good to get stuff from a stream, but then let it dry completely before you then submerge it.
Correct. Yeah. Yeah. That was, we talked about that, kind of just speculating on why that is, but uh, it seemed to work because immediately after the day after the first class, and he talked about submerged material is really, really good. Uh, I immediately went and did like four cultures from submerged material and just let them not dry out, and I got nothing out of them. But after the next class, he was
Mm.
like, "Yeah, you need to let them dry out." And from there, I saw a noticeable improvement, in immediate plasmodium.
I wonder if that's
Plasmodia.
synonymous to shocking, like shiitake logs, synonymous to like a cold
Hmm.
soil, because that'll stimulate the mushrooms to pop out with the cold soak. Um, but
Yeah, not a bad theory. I
yeah,
mean,
I thought.
that's
So
kind of what when I say we were speculating it like that. These are the type of things we all threw out, and ultimately, Dr. Stevenson was like, "I don't know why, but it works."
so you collect it in the field, you take it home, you dry it out, and then you open it up and you flood it, and then you leave it in a dark space.
Yes,
So
and
how do you know that what fruits on the leaves came from the stream and didn't uh attach to the substrate in your apartment or when you make the culture?
yeah, that one's out of my. Uh, I don't know the answer to that, Jason.
When if a somebody who studies these things frequently, opens it up and then floods it and gets a 90% success rate, he's probably got spores floating
Yeah, right. In
all over the place.
whatever workspace he's working in. Yeah,
Yeah.
it's it. I mean, That's also a good theory. I'm not sure.
Cool. Thanks, Brandon.
Yeah, no problem. Thanks for looking at it.
All right. Hunter, I got your next three up.
Wait, I just want to elaborate on what Jason, were you suggesting that like the, these things might grow in Brandon's bathtub or something if he closed off his, like flooded his bathtub, closed off his bathtub and kept it dark just because they're living in his house?
Or just the spores, being airborne in his apartment.
Um,
Or,
okay.
or I mean, the dozens, if not hundreds of slime molds that I've brought home in my backpack that backpack, funny enough, I do a lot of this work in my bathroom uh, because I can clean surfaces really easily. Um, but that backpack is sitting in the bathroom with me. So,
Yeah. I mean, you could have slime mold spores permanently embedded in your lungs, and every time you exhale, contaminating the sample. I don't know. I don't know how these things work and how persistent spores and propagules are. But I mean, it seems like in nature they'd have to be to get to where they're going. I mean, for the frequency that I see fruiting slime molds in the wild,
yeah.
90% success rate on culture seems surprising because you don't always see slime molds out there fruiting,
Well,
actively
I,
releasing
I,
spores.
I guess there is uh, counter evidence to that, Jason, because Dr. Stevenson collects. I mean, he's collected from all over the world. I mean that. I mean, he's collected in Antarctica and found new species from cultures. So,
Right,
he didn't bring that. That wasn't in his house. You know what I mean?
right,
Um, similar stories from New Zealand and Australia. Uh, he wrote the monographs for both New Zealand and Australia, discovered new species, and those species were discovered in his cultures. He didn't collect them in the field. Um, and a lot of mixomycetologists will say that there's an easy way to study mixos and there's a hard way to study mixos. The easy way is through cultures. The hard way is collecting in the field. Uh,
right.
so I don't know if that means I think that is kind of disproves the how much of this is in my environment versus in nature, but you know that's also
Well,
anecdotal, I
well,
guess.
but both can be true too, though. You know,
Sure.
how do you know that you've got a sample that is pure? I'm thinking especially if one day we have um, a list of threatened and endangered slime molds in New Jersey, and you do a culture, you find it on a site, you gotta legally prove in a regulatory set, fashion that it came from where it came from. How do you do QA, QC and demonstrate that, culture is not contaminated and that it actually came from the environment? Probably, you could happen 95% of the time, but how do you know?
Yeah,
Yeah,
it's fair.
it's such a different process than, say, like um, culturing mushrooms from spores, where it's all sterile, the agar, and every aspect of it. You're using a flow hood, and trying to keep any other elements out. Um, so but that's what's so fun about it is like I like the surprise aspect of this.
Yeah.
You just don't know what you're gonna get.
Every morning is Christmas morning.
Plus you get,
Yeah,
plus you get to chase them around and find where you hiding that this morning. It's fun.
as they move. That is so cool.
All right, you want to go next, Hunter?
Yeah, I would love to. Thank you. So yeah, I would have shared these, but then I thought I saw we were doing lichens, but this I found in a bottomland hunting for morels, and I am pretty sure I found the same mushroom and showed it last year. It's really really pretty, um, and I mean I'm noticing it almost has the current kills, um, and um, which is interesting. And I mean, I guess Inat is probably right. The genus has changed. Um, this is Xeromphalona, or is that what it was? Starts with an X. Oh gosh. But this used to be. Dave has showed it a couple times. The smaller, is it Xeromphalona? Uh, oh gosh, hold on a second. Um,
Here it is. Yeah.
Are you thinking of
Xeromphalina.
Xeromphalina?
of what?
Oh, yeah, there it is. Xeromphalina.
Yeah, Xeromphalina. So there's the Campanella, the little one, and then this tenuipes. And I have a picture of this tenuipes from Wisconsin that looks totally different. Tenuipes looks totally different from this. Um, and actually, I'm looking at Mushroom Expert, and they don't even. Oh, they have two other ones, Quaticinalis and Kaufmannii. Um, but does what do you guys think of this? Are you familiar with it at all? Or? This would be deciduous wood, maybe sugar maple, maybe sycamore, maybe black cherry, degraded wood that it was growing on. But it doesn't really look like the pictures of tenuipes to me. And I know it's
Yeah,
not
doesn't
Campanella.
doesn't Heimiomyces have like a translucent dark stipe?
Yeah, everything's wrong about it. It's not. It, um, yes, I think it looks very different. Um, the stipe is described as, well, thick, more or less equal, dry, densely and finely fuzzy, golden orangish, darkening to reddish brown or brown from the base upward, fairly tough.
Yeah, I've never found this species is one of those I talk about it all the time. When I'm out of ideas, I'll go to the AI on iNet, and this one pops up so often for things that I
Mm,
don't I don't think look anything like it. Um,
okay. Yeah, I think it's gotta be. I mean, I would say like, well, the only thing is the decurrent gills. That's when I started buying into it a little bit, because Campanella has that. This doesn't, but Xeromphalina campanella has sort of
Well, Xeromphalina, Capanella, and what's the other one? Is it Schwartzii?
Kaufmannii.
Kaufmannii. Um, they're very small and
Yes, right.
look, they're
This is big.
yeah. Xeromphalina, I think I know pretty well. You can pretty much work those two species out just based on what kind of wood it's growing on. Now, I assume this was moved out of Xeromphalina, probably. I mean, just morphologically, maybe. But yeah, I don't know. I don't know what this thing is. Long story short.
I mean,
Yeah.
it looks like you're saying that yours had the current gills? And those look like the like depressions from the
Right. They look
gills.
notched with what do you call it? Igor, help me out. What do you call that?
Can we get that?
Striate more
Subdecurrent?
what
subdecurrent.
the gills?
Well,
Yeah,
how the stem looks like it has the current gills up near the top.
But you
Oh,
see, then I get the current, they sort of go up and then they go down. So it's almost like it's notched, and then there are these ribs coming down the apex of the stipe. I've never seen anything like that before. Because the current gills, they only go in one direction, top to
yeah.
bottom. Okay, they don't
You know
change
what it
direction
reminds
here.
me, Igor? You know what it reminds me of when you see that at the apex of an amanita, right? Which is it's not the actual gill structure. It's kind of like as Chris mentioned, it's like depressions almost from the gills, if that makes sense.
Well, the imprint, because remember, in Amanita, all the structures are sort of permeated by the vulva. So things have to separate and tear down, tear apart from each other. And that's why what's left, they're not impression of the gills, but impression of the material that was on the gill edges that got left on the stipe, if that's what you're referring to. That's different.
Yeah.
These
you know,
things don't, yeah, these things don't have these kind of tissues that amanitas have.
look at the young one too, right there. Like it's staring at you. It doesn't. That is not as prominent of a feature.
Yeah, but if you look on the left, they kind of do look the current, but they change direction and sort of go up and then they descend
Yeah,
because it's probably some kind of a umbo on the cap or whatever. I mean, if you just compare that to say chanterelles that are the current, you have this shape of the cap, sort of it's lifted upwards, and the gills sort of go, they're horizontal first, and as they go toward the stipe, they begin to go down, and they just run down the step. Here we have things go up and then down. But yeah, I mean, I think this is probably qualifies as a current gill, which
I
is very
tried
neat.
to get a spore print. I couldn't get anything off of it at all. I don't know why.
I mean, you're saying this is Xeromphalina? They kind of look robust for that, right?
I think so. I mean, that's just what INAT said it was.
How big were these things? Just out of curiosity.
Big, like 3 or 4 inches tall.
Wow. Well,
Do we think maybe
I guess I
do,
know nothing about Xeromphalina other than the Campanella and Kaufmannii.
What about,
do we
go ahead.
think maybe there's some partial veil material on the margin of the cap here?
Partial veil. I didn't see any partial veil.
I see what you're saying. It like a little overhang.
Yeah.
I don't.
Yeah, I don't think this would be a something that would have a partial veil. If so, fleeting. Obviously, there's no remnant on the site.
could be just, sterile margin, which is very narrow.
Yeah.
Gills don't quite reach it, so there's like a fraction of a millimeter there, sort of. I don't know. I mean, if there's partial veil, I mean, there should be remnants of that on the stipe as well, typically, right?
Right.
I would.
I
I
don't
mean,
know.
my first guess would just be something like gymnopilus or gymnopolis, or by the color.
Yeah, the reason that I'm trying to talk myself into that margin having some material there is the caps kind of reminded me of Hypholoma.
Yes, but the gill color's not right, right? Or maybe.
Oh, I don't know. But but again, that would show some, remnant on the apex of the site somewhere.
I missed discussion earlier. Was there a spore print?
I tried. I could not get any spores off of it. I don't know why.
I mean, they
Nothing.
kind of look mature, so why wouldn't they drop spores?
I know. I'm not sure. Maybe it was refrigerated or something.
Hmm. Well, there's always sequencing. uh,
Yeah, I don't think I can come up with a little cap I brought home. It dried up, and I don't think I can say with certainty it is it is the cap of this mushroom.
what happened to the other two mushrooms?
They stayed there.
Oh, you didn't take them?
No.
Hmm.
I just took one.
Oh, well.
All right. Yeah. Okay.
Is-
Well, we can move on. I have other stuff. Chris, I watched your microscopy demo. Is really good and
Does
helpful.
anybody recognize this species? Is this a possible candidate?
Hmm.
Looks like it's got kind of decurrent.
Well, the photograph on the bottom looks kind of similar. That shows like lots of caps. Yeah.
Yeah, and I mean they were growing cespitosely, correct? And I think
And these look different. Or maybe not. There was one photograph with a lot of fruiting bodies showing caps. And those kind of looked similar to what Hunter has. Yeah, maybe this one. These almost look like Hyphalomish, the caps are, right?
Yeah.
very much so. Yeah.
So, is this from this continent or?
Is this a after fire mushroom?
No, it's
Oh,
either
wow.
end.
Stark
The
absence in
center.
the middle, huh?
Yeah.
Is this one that grows after forest fire?
I don't know.
And they have that in Europe as well, huh?
It does have a real similar look.
Pyrrhulomyces, astragalinus.
And this was observed in April.
Yeah. This was just 10 days ago or at the max, at the most.
Yeah, it's a weird time of year.
Not even that long ago. Yeah, it's very warm here, though. I mean, we're jumped ahead.
Yeah.
Boy, it's an old species. It's a Frisian species. known as Agaricus astragalinus.
It's funny because I feel like I showed the same thing last year and found it in the same place, but I don't think I have a INAT entry for it. I think I just shared it.
Growing on conifers.
So there's no conifer here. For sure, none.
But the species that Jason suggested, the Pyrrhulomyces astragalinus, says here it's growing on decaying wood, primarily on conifers, logs and stumps.
Looks similar to this, but it's not panning out the range, time of year, substrate.
And that's in the family Strophareaceae. Pyrrhulomyces, that is.
That's Typhlomas right?
Pardon me?
Typhloma's in that too, right?
Probably. Let me take a look. Agrasibi, Bagbodia, Deconica, Hemifoliota, Hyphaloma, Curomyces, Loretomyces, Foliota, Protostropharia, Stropharia, and Wararora, and a few other ones that I've never heard of. So yeah.
Did you say is Agrosophy in there?
Yeah, according to Wikipedia, Agrasibi is in Strophareaceae. Yeah.
Right. And what's that chip cherry one?
Bagbodia, Broniella, Deconica, Hebelamina, Hemifoliota, Hyphaloma, Cuneramyces, Loratomyces,
Loratomyces, right? Yeah.
Loratomyces ceras. Yeah, like right. Pachylepyrium, never heard of that one. Theogalera, Theonematoloma, Foliota, Protostropharia, Pseudogymnopolis, Pyrrhulomyces, obviously, Stagnicola, and Stropharia, and Wararora, sorry.
Pseudogymnopolis.
Pseudogymnopolis, yeah.
That's what I was laughing at. Pseudogymnopolis.
Pseudogymnopolis. I wonder where that name came from. Let's take a look at Index Fungorum.
Probably because they didn't like Gymnopoloides.
It's a fairly new genus, 1974. European, apparently. Yeah, weird.
Well,
It's
I
a
mean,
nice yellow color.
it's
It
beautiful.
it sort of reminds me of Tricholomopsis a little.
Yeah, that my head went there at one point as well. Like a Tricholomopsis or Flammula, Tricholomopsis, something like that.
All right. What else we got?
Oh, so I collected Neogyromitra brunnea again, and this was actually a quarter of the picture we just saw. There were like four clustered together, and I did the microscopy, and this time I was able to, after watching the video that Chris gave, I was inspired and finally got some spores off this and acai as well. And so that was pretty exciting.
They get this beautiful purple color.
That's that flaxing, using too much of it though. So I still need to figure out how to control my amount. But I think I know how. I just need to drag it or something, pull it with paper towel from the other side into it.
Yeah.
You get the dye as a liquid,
Yes.
and then do you dilute it in like a small beaker?
Um, Igor, should I be diluting the flaxine? I think it's already diluted somewhat.
Well, if it's coming out too strong, then you can dilute it to make it.
Okay, just with water or
Yeah, that's what I
okay.
would do.
Okay. Well, I mean, I tried putting a water drop and then adding a flaxine drop, and then it was still pretty intense.
Hmm.
But oh, there's some.
Yeah, if you keep adding, water from one side and then, kind of just paper towel in the other side and just keep like dragging it through, you'll wash a ton of stuff out.
Okay.
Yeah, and I think I don't know about with when I get dyes, I was under the impression that they're intended to be diluted because a small bottle will only last you so long if you use it directly on the slide. So if you fill a small beaker with water and you put a drop or two into that, then you can use that to do your mounting, and it saves the bottle from running out as quickly.
Plus a lot of you don't have to do all that dragging. I like that it's
Right.
and I think I will try a small diluted quantity and see how that goes. Um, and if that doesn't work, then I'll go back to the try dragging. But yeah, that sounds like a really good way. So yeah, I was super excited to get some halfway decent microscopy. And
Yeah.
yeah, I mean it's okay. It's cool. The spores are cool, and I wish I could. I know we were looking at some of the other old Gyromitra group, and so I wish I could speak to how they differ, but um, I can't at the moment. But this is our very common around here. I mean, and there were about probably around 30 of them in this one spot where I found this. And as I was saying, it was actually it's it's a it's a cluster of them. There's like four growing out of a central location, all mashed together. So I just in the other picture you can see just like a quarter of that group.
Hmm.
And the stipe is super white. Yeah, so this was a piece of that, like, but it was sort of had its own stipe.
Cool.
Yeah, it was neat. And these are now considered edible, and people are eating them, but I am not.
Hmm.
That's the thing I always had learned about the Gyromitra was that the toxin level that, that could really vary from location to location and specimen to specimen. And apparently it's been, enough of a sample's been taken that's been debunked, but you never know. Maybe it's true. You might hit the right spot. I don't know.
Yeah.
I don't want to find out, but a lot of people are eating them. So, and then I've got Verpa conica, and I showed this last year. I found it again in the same spot. It's considered a morel, the Verpas. And their edibility is kind of questionable, but they are also now it's said that they are edible and just should be cooked thoroughly, but I also don't eat them. They're sometimes called the thimble cap, and the cap attaches at the very top of the stem and separates very easily. It just sits right up on top. It's kind of interesting. It's a neat looking mushroom, and I did find the spores, which are very neat and clean and regular.
So I don't think I've ever found a Verpa. They call are these half-free morels?
No, half free morel. The cap attaches. The
Halfway.
attachment, yeah, it's halfway up the cap. The attachment. So yeah, and those tend to have um lines down the cap. I haven't seen many this year, but I'll try to get a picture for next week. That's Morchella punctipes, and those also have little dots on the stipe, which is what they were named for, the half free morel. So no, this would be like a fully free morel.
Cool.
Yeah.
So that connection is up right, where a traditional gill attachment would be. Obviously, that's not the situation here, but it's up at the apex of the thing.
Yes. Yeah. And last year when I showed it, I showed the separation, and it's pretty interesting. It just sits right on top. Yeah.
Cool. Thank you.
Yeah.
Yeah, and I don't see any records of this on iNaturalist for New Jersey, and the Mycoportal just has a single collection from 1948 up in Warren County. So yeah, we might be on the far eastern edge of the species if we have it at all.
How about can you look up punctipes, the Morchella punctipes? Do you guys see that I'm in South Jersey. I don't get up to North Jersey all that much, but I don't know if people see that down south. I don't think I've ever seen that one either.
Yeah, let's look at its range.
Further east, it comes all the way down into New York and not much in between. I might be sampling bias because there's so many people in New York and Philly.
Yeah.
That thing's funny looking. It's like wearing a morel hat.
It's a Shihong. Yeah. It's one of Shihongs.
Yeah, and that looks the part, right?
So we're looking at punctipes.
Yeah.
Yeah, I found punctipes in my woods here on my property, but that was only once and many years ago. And we also found this, I think, in Morristown area. I've seen them too.
So it's probably here, but more infrequent in South Jersey.
You can really see the points on the stem of this one if you look really carefully. Can you see all those little dots?
Yeah.
Yeah.
They're like hairs almost, like, right? Like stubble.
Yeah. Mm-hmm. And that's why it got the name. But then Verpa bohemica looks very similar too. We should bring that up too, because they look really similar.
Do we have bohemica here in North America?
Yes.
Oh, okay.
That one, I think they look more like a true morel.
You know what I just saw in Mushroom Observer? There's this user with a very high score. You probably know her, Hunter. Eva Skific from Canada.
Oh yeah, I'm familiar with who she is. Yeah.
Yeah, so she posted Verpa bohemica, and I was like already thinking, do we even have that thing in North America? But up maybe like in the boreal area, it's like a northern species.
Yeah, we do have it, not a ton, but I see it throughout Illinois, I would say, even in southern Illinois.
Yeah, so Jason is showing bohemica right here on the map.
Yeah.
Yeah. And worldwide, with the exception of the West Coast USA, seems to be yeah, it doesn't go south of
Yeah.
this line.
So
Yeah,
do you think that a lot of these northeastern identifications are wrong, or all of them are correct, bona fide bohemica?
I think
so
it's tricky to
Can
tell.
you click like on some of these just to see if they kind of look like bohemica as opposed to punctipes?
Yeah, I think these do all look like bohemica. I mean, I don't think we saw enough samples of punctipes personally for you guys to really get the difference. And I think it's the attachment of the cap that is one of the big differences and makes it look different. But clearly these don't have those points either on the stem. Yeah, see how the girl has them on her fingers? Because, well, of course, you could do that with a morel too, but
Yeah,
because they were attached up at the very top.
I'm thinking, we used to call the mushroom that is now Morchella punctipes, we used to call it Morchella semilibera, and that's European. So that one is not in North America, right?
I'm not familiar with it. Can you scroll down? Because I did see one on the left that did look like punctipes to me. This one with the bright green, the one that's like out of, yeah. Can we
Yeah.
click on that?
And there's another one in the same row, I think third from the left, that also look like punctipes to me.
Yeah, I think this may be punctipes.
Well, somebody crossed it out.
it's the same guy. Changed his mind.
Yeah. I wonder why.
Yeah.
Which was the other one you saw?
Yeah, the third one. Yeah, this one.
Here.
Yeah, I thought this one could have been punctipes.
That one has a very smooth stipe.
But it's old. It's
True.
well past its prime. So yeah, it's going to be questionable. I mean, you could be right. This is bohemica.
And another difference is when you cut open the conica and bohemica, it's got a pithy interior, whereas the Morchella punctipus will be, I'm pretty sure will be completely hollow like a morel, a true morel, because it is a Morchella. But you're right.
Yeah, all these cottony stuffing in there, right?
Yeah, and the Verpas.
I mean, you can see the shape of the cap. I mean, this is not punctipes or semilibera. Those have a specific shape of the cap. Bell shaped. These are not that. They look funky.
Like a thimble.
Yeah, exactly. A thimbleful of spores.
Yeah. Well, you guys should come out and come out during morel season if it's dry next year or even this year.
So it's not even that boreal. I mean, there's a lot of observations from Pennsylvania, right, Jason?
Yeah, a few.
Yeah, a few, and yeah, southern New York. But it tends to be more northern, so it would not go beyond a certain. It wouldn't
There's
go,
a
south of the Mason-Dixon line. That's that.
right.
It's that borderline that I always like to mention like 1,000 times. Mason-Dixon line, magic. It's the cutoff between southern and northern mushrooms.
It looked like there was a Plischke bohemica.
Yeah,
So
a few observations near Pittsburgh.
I love how you use the Mason-Dixon line. I
Oh,
love
Mason-Dixon is lovely. I mean, it's like magic. It works all the time.
yeah, it's like the east or west of the Rockies.
Yeah. Yeah, this is Verpa bohemica. And this is Plischke's, right? Yes, it is. Yeah. Mm-hmm.
Yeah, and you can see that cap attachment up at the very top, as opposed to like a true morel would attach at the bottom of the cap, and then the half free, the punctipes would be like halfway out.
Right. Yeah, this is completely unattached on the bottom.
Yeah, that's cool.
Yeah, and you got the cottony stuff in there, right? So the pithy, fluffy material.
Is there any information on this post, Jason, about where this was found? It looks like it's in a lawn.
Not necessarily. Plischke, John likes to pose his mushrooms on a bed of grass or moss or something like that. So that's
And
what he did. I'm pretty sure he's staging this.
yeah, this is an old observation too. This is pre-INAT. He was just going through old
Oh,
photos and posting
Oh,
it.
one
my God.
25-year-old picture.
These do tend to grow, in the same areas you would find morels.
Cool. And then we've got Morchella prava.
Mm-hmm.
Yeah, and this I've shown past years to you guys, and I found it again in the same place, exact same place, looking pretty much the same, which is like a giant tree trunk of a stipe, and then this large cap that looks really like a very young morel. But I'm pretty sure it's Morchella prava. I've dried it, and I'm hoping to get it DNA tested. I'm gonna work on that. Unfortunately, I couldn't get any spores. I think it was immature. This is from the ridges. I scraped them, and this is what I got. And then I tried to scrape the pits to get the spores, and I got nothing. What is this?
Well, I don't know, but I still think this could be either Ulmaria or Americana. I think Prava has a very misshapen cap. Yours looks a little bit more sort of uniform, but I could be wrong. Like I said, Prava is that species, a funky species from the northern tier of the United States. We don't have it here in Pennsylvania or New Jersey. But if you look at the photographs of Prava, it has a certain look about it. I think this just could be a Americana.
Well, my picture from the last year, it was the pits were more open, and it looked more similar to some of the other observations. But can we look at the other picture of it? There should be another. Oh
Yeah, I'm
yeah.
also going to post a link from Mushroom Expert. I think it's a Mycological species. So he has some photographs on his website, so you can take a look
Ev-
there.
Even the stipe shape, I mean, is like, look at the ones on the right, on the bottom, how wide that middle one gets at the bottom, just
Here?
like mine.
Oh,
See,
What I like about prava is that the gaps are these cells are misshapen and the edges are very thick and
right.
much lighter in color and are brown, not gray. Can you click on these photographs? Yeah. So you see,
I think these are just like older specimens, though.
hmm. Like I said, I'm not an expert on morels. I sort of give up on them in the sense that the papers are there, so you can educate yourself about the species and the names, whether they're legitimate or not, and phylogeny and so forth. But if I cannot really ID them well in the field. I mean, they're very mutable and just impossible. Except that now we have fewer species because of all the trees are gone. Ash and um, what is it?
Al-
Elms, yeah, they're pretty much gone. So there's going to be a diminution, in number of species.
I was wondering if it could hybridize, because that's what I was thinking. I'm like, oh well, it doesn't maybe look as much like Prava this year. Could it have like hybridized with Americana?
I don't know if they can hybridize. Don't know much about hybridization in the context of morels, but Polites a little bit. Like Lexinum did that a lot early on before everything split up. That's why its DNA is so kind of useless. ITS, that is, because there was a lot of introgression and hybridization early on in that genus. But morels, I have no idea about.
Well, I'm gonna find my picture from the prior year and share it with you guys. Because it does look, it's more open and more sort of geometrical. The pits in a
Yeah,
very unique way.
yeah. I mean, the best way to sell this argument would be obviously a sequence.
Yeah,
If ITS resolved, you have to check if ITS can resolve prava from
right.
Americana, because I think for some uh, black and yellow morels, ITS doesn't work so well. I mean,
Yeah,
it's still a decent.
Hey, Jason, can we scroll down to see if this has a season, even in the more northern states?
this right here, April, May, June.
Hey, Brandon, do you collect or have you ever collected morels in the Upper Peninsula? I think you have relatives in there, right?
Yeah, I've never made this season,
Oh,
which is why I'm asking that question,
I see. Got it.
because I plan on going up there at the end of June, and I'm wondering if
it'll be
they're more.
over
It'll be over.
all
Ah,
over.
Yeah,
Yeah,
looks like it.
especially this year. But I mean, you might get them in like the first week of June then, but no. I put in the chat mine from last year from the same spot, and you can see. I mean, maybe I didn't find it again, but and I just found it last year. But if you check out the one in the chat, it's got this weird pit shapes.
No, it's tried to
And
play. It doesn't. It won't let
Yeah, I
me
mean,
open it.
I'm not trying to convince you otherwise. I'm just saying that, kind of playing a little bit of a devil's advocate. That these things are sometimes, uh, you look at these species and you just get confused. I thought what I was collecting was Morchella septiformis, and that happened to be like another, morel, septentrionalis, or I forget whatever the species name is. But I had to sequence it to get the name. But there was something strange about them too. So yeah, I mean, sure. Yeah, I mean, I'm not saying this is not prava. I mean, it could be prava.
Did you see the one in the chat?
I'm looking at it, but I cannot blow it up. But I think I can see it pretty well. I mean, the one that you have
Yeah.
in the chat looks more like prava to me.
Yeah.
Yeah.
But see how the stem is the same stem shape, and I found it like right in the same spot too.
Yeah,
But
but I've seen this in the little yellow morels as well here in my neck of the woods. The flaring of the stipe on the bottom and all these wrinkles and ridges and holes or whatever, pits. I've seen
Mhm.
this before, so I don't think this is like universally a universal trait. I mean, it's a universal trait. It's not a specific trait for any specific species. That's what I'm trying to say. So I don't think it's a good morphological character. What you see on the stipe. I think the caps, I
Okay.
think, more probably telling than the stipes.
Mm-hmm. Okay.
Yeah,
Well,
these
let's
look more like prava. So yeah,
Here's the mycoporal Brandon for prava. You can see the dates here. Most of them are May. Like the latest I see is June 8th. That's California.
Yeah, and yeah, that's good info. I see all the Quo ones from Michigan at the top there, and those are earlier dates, it seems.
Yeah, if we go back and just
cool, right?
Because
Michigan.
I'm sorry. Go ahead.
well, I was just, when I say the UP of Michigan, I mean the we often go into town, and that's in Canada. It's like it's way up there. So I'm wondering if that seasonality, uh, might
well
be a little
now
bit later.
Yeah.
now
Well,
having a morel foray in British Columbia. Do you know those dates? Have you seen that?
No.
So let's see, here it is. It's June 1st to June 4th in British Columbia. So that's for the Vancouver Mycological Society. So that's probably, the very latest you're going to find them in, even in the UP, I think.
maybe if the weather, the winter was cold and it's still probably very chilly up there, so you may have an extended season this year because of that, especially if it continues to be below the average. So who knows? I mean, Kukua writes here, growing gregariously under hardwoods and conifers in sandy soil near bodies of water, April, May, and June. Apparently widely distributed in northern North America. Confirmed collections from Montana to Saskatchewan, South Dakota, Michigan, and Ontario. So this thing likes to be up north, really, really up north.
Hmm.
Cool.
Cool. Okay. Well, I better get it sequenced so I can know, except that might fail me anyways. So,
Yeah, take a look at the whatever papers you can come up with. I think most papers are available online for free. See if ITS is a good barcode to separate prava from away from all the other yellow morels.
okay.
Because if not, then you'll be just wasting your
Right.
money and time. So
Yeah.
you may
Okay.
have to go for a different gene or locus.
Yeah. All right.
Cool. Thanks. Thanks, Hunter.
Yeah. Thank you guys.
So, Lila, you said you had some to share?
Yeah, I've got some lichen. So
Cool.
let me try to do this. Get it up. All right. Okay. All right, can you see this?
Yes.
Mm-hmm.
All right. Let's start with this one. I don't think this is necessarily the right ID, but I thought this was an interesting
That's actually a lichen.
thing. I think
Definitely a
it's
lichen.
it's on rock.
Porpidia would be more flat, right? And I'm asking that as a question to Jason, not like knowing that.
Well, there's Porpidia, the most common Porpidia albocerulorescens, which has more flat apothecia, but I do think that macrocarpa has those more domed shaped apothecia.
Cool.
Guess this
Thank you.
first picture is the best one. Um, but yeah, so that's what I'm
But
calling it.
the one thing that makes me question it is you see how it don't look at the apothecia, look at the thallus, the white stuff. You see how bumpy it is?
Yes,
Usually Porpidia macrocarpa is smoother. It doesn't
smoother,
have that
right?
bumpiness. Um,
So would you call this one areolate, or is this just kind of whatever it is, Soridia, Isidia?
no, it's definitely not ceridiate or isidiate. That's the thallus. Those aren't asexual propagules.
Ah,
Um, but areolate is usually more irregular, and this is bumpy. It's not like cracking up into subdivisions like a dried mud, where it kind of cracks.
yeah, that makes a lot.
This looks
Yeah,
like,
that makes sense. These are like mounds, right?
yeah, this is an aggregate of mounds. Um, So yeah, I would need to look at the spores for this one to get any further and put it through the
Okay.
keys.
Okay. Do we
But
think it's Porpidia for a genus or can't even say to that level?
No,
Okay.
in fact, I'd wager that it's not a Porpidia at
Not a
all.
Porpidia. All right. I'll change it just to,
What is well,
oops. Yeah. All
yeah, so I
right.
know that with the app, if like go to your second photo, um, like I don't know how the iNat AI does it, but the first picture was really good because you saw like that side view and it was a really close up photo.
Yeah.
But I would have put that as the second photo and made this one the first one because like when most people add an observation, it's more likely to be from the perspective of this.
Uh-huh.
So I'd want the AI to compare this photo to all the other photos, not the first one.
Yeah. I might have done that and then just switch the order. I often run when I don't know what it is, I run all my photos through the iNet.
Yeah, I know on
Yeah.
the app you can choose which photo you want to
Yep.
use, but I don't know if the website. I think the website just takes the first photo. I'm not sure.
I think the website, if you cycle through those, uh,
It'll give you different answers.
yes, I think so.
Yeah,
When you
it definitely does
when
that. If you have different views of things, you can get different recommendations.
So click on the second photo, and I'm just curious what the AI suggestions are if you see down below. It's not Buellia. Lecidella might be a possibility.
hmm,
Yeah, that kind of.
that doesn't look entirely dissimilar, does it?
Yeah. But a lot of these things you really need spores, and in order to get the spores, you gotta slice those apothecia into a section, put it on a slide.
Okay.
You can't get a spore print out of a lichen. You gotta section it.
You got to work at it. Yeah. Tear it up. Okay. All right.
Uh,
That's,
is, is Buellia or are those apothecia ever going to be that raised or mounded, or you know, or are
They
they going to be?
can on a Buellia, but I think most, if not all of our Buellia occur on wood or twigs.
Oh, right.
Yeah, and even the thallus looks different there
yeah.
than the one that Lila had.
Okay.
Yeah, because often when I think I find a Buellia, I rub my finger like along the thing, and yeah, it's on wood. Um, but I, don't get a noticeable raised bump. But those apothecia seem much smaller than the thing we just looked at, maybe.
Mhm. All right, so this one is likely in the genus Xanthoparmelia. And we've got four common species of Xanthoparmelia in New Jersey. And the two distinguishing features are, one, are there isidia or are there no isidia? Sometimes it can have apothecia and isidia, in which case you would call it isidiate. I don't see any isidia here, so this would be one of the two apotheciate species. And then the second trait is the color of the underside of the lichen. One is kind of like a pale brown and the other is black.
Okay. I think,
So,
let's see. I think this one, I have a picture of the underside. I think this is the
mhm.
same fruit body, but did I get an underside? Yeah, here's the
Nice.
underside.
Yeah, so that looks dark to me. So that would make the Xanthoparmelia angustiphylla.
And when you say dark, you mean not black. So like
Yeah,
brown.
like a pale brown. Like a black is going to be, yeah, maybe this is the pale brown. It's kind of hard because it's wet. Um,
Yeah. Here it's kind of brown here.
yeah, I mean black. Um, though, yeah. So that would make it Cumberlandia, I think. Let's see.
And then I think this is where I tore it. Is that the medulla white on the inside? Is that? I don't know. It's been a while.
Yeah, yeah. Looks like the medulla. Let's see if I could find.
Yeah, and I only asked that to try to figure out the difference between brown and black on the underside of these things, not to like challenge your ID.
Mhm.
Yeah.
The I thought this was interesting how this was very vertical here. I don't know, that caught my
Yeah.
eye. Maybe that's nothing
It's really,
unusual, but
really trying to stretch those spores as far as it can.
mhm. Holding water.
Yeah, that's a good shot.
Okay. All right, so that was those. Then all right, so here's one that I thought was a Xanthoparmelia.
Interesting.
Yeah, those apothecia are huge. Is this in New York?
This is in New York on a
Hmm.
rock on a stone wall.
Hmm.
So that one on the bottom, you see how it's kind of red and it's got black dots on it?
This part here.
Yeah. And below it, on the same structure, you see those black dots on it?
Something. Why is this on my screen? The controls are on my screen here in the way.
Yeah, I hate
There
that.
we go. Yeah, oh yeah, the black dots down here.
Yeah, that looks like a Lichenicolous fungus to me, like that's infected, and those black dots are the apothecia of the parasitic fungus on the lichen. So apothecia on an apothecia.
Huh. Okay.
So then you gotta section those little suckers.
Yeah.
Yeah,
Are,
I guess
are they
Yeah.
kind of tipped
the
over and
the
stipitate?
lichenized apothecia, you get an appreciation like, oh, that's way more easy to work with.
Yeah.
Yeah.
Yeah,
Yeah,
there's no
start with the Lichenicolous fungi and then lichens will be a piece of cake.
but yeah, I thought this was quite beautiful with the crusts or whatever around the edges there.
Yeah, the thallus material.
Mhm. Yeah, I don't think got an underside of anything here.
Was that the same spot as the other one?
Yeah, it's probably along the same rock wall.
Yeah, looks like it's all. Zoom in again on that one. You just see. I mean that. I mean the apothecia looks similar, but the I don't know the thallus doesn't look as foliose. I mean it is foliose, but I don't see the distinct lobes as much as I did on the other one. Which is kind of odd, but I think it's all probably Xanthoparmelia cumberlandia, which is the apotheciate one with the pale underside.
Cumberlandia.
Yeah.
Okay.
And
So
then
the
what about those
oh,
3 or 4 apothecia kind of below the big brown ones? The seem to be a couple black ones with a mealy rim. Is that a
These over here.
yeah?
yeah,
Is
those
that
are
a
just young.
okay?
Yeah.
Yeah, that's what I
Yeah.
was wondering.
Yeah, I
Cool,
think there's just all kinds of different stages
Yeah.
here.
nice, cool find.
All right. Uh, all right. so this is probably wrong. This, I think, was a INA recommendation, but also on rock.
Yeah, but this one's Isidia. See all the Isidia?
Yeah.
So that rules out Angustiphylla and Cumberlandia. So now we've got between Plidii and Conspersa. And again, you got to see if the underside is pale or black, pale brown or black.
Mm.
And if it's pale brown, then I think it's Plidii, and if it's black, then it's Conspersa.
Are there any chemical tests that can tell those two apart? If my eyes can't tell the difference between brown and black?
Let's see. Well, you can do between Conspersa and Plidii. You can do a K test on the medulla. One's going to be K plus yellow, and the other is going to be K plus yellow turning dingy brown.
That's another distinction that my eyes can't really tell the difference between.
Yeah.
But cool.
Very cool.
Yeah, it was fun. All right, one more. I think this is somebody confirmed this. So I don't know if this is right or not.
Yeah, that looks right to me.
Okay. But also quite beautiful.
Yeah.
All
That
these
looks
things are
like underwater coral snorkeling or something.
something, yeah.
So neat.
So I'd like to ask about this one. If we can, Lila, do you have a photo of like a good photo of the margin? I mean,
Margin.
we can see it pretty good here.
Yeah, this is probably the best spot right here. Not
Is,
not great.
is there a way that's how you tell, right, Jason, between this and Stellaris, I guess would be the other one that you would compare it to?
Well,
Just whether
Stellar, you
Quite
see all
different from
that.
Stellaris, I think.
Yeah, you see how it's got all that crusty, I don't know if those, I don't know what all of those structures are in between the apothecia, lobules or something.
yeah. Okay. Yep.
Yeah, Stellaris won't have that. It's also it's hard to tell from the photo, but millegrana is also much smaller. Like those lobes are finer. Like everything about it is. I mean,
Tinier.
this whole thallus here could be smaller than a dime.
Cool.
Yeah, I think it was quite small. And I think right here I've got Stellaris,
Yeah.
quite a bit different, I think.
Yeah, so I called a lot of these things like I would have called this millegrana or millegrana a couple years ago, but I think is Stellaris the more common one, or which one is more common, Jason?
Well, um it's it's more common to see Stellaris because you can see it from a distance. Usually only see
Gotcha.
Millegrana if you're up looking on a bark. Millegrana also kind of likes smooth surfaces, and Stellaris is almost always on canopy branches. So Millegrana can be on both, but if you're looking on the trunk of a tree, you're more likely to see Millegrana, and Stellaris is going to be on branches that fall.
Yeah, yeah, I always see it on like oak sticks, fairly small oak sticks.
Yeah. And we can call it all stellaris, but there's also pumilior, which they're very difficult to tell apart. So um, I know there are some people who are working on the DNA between the two of them to see exactly what's going on, and they're hoping they'll be able to lump them into one species. But telling them apart really, I think, requires thin layer chromatography.
Mm, okay.
Cool. Thank you,
Yeah.
both
Uh,
of you.
All right, so I think that was most of them except for this. Oh, well, all right, real quick, we'll look at this orange one. Um,
Yeah,
on a rock. Really quite tiny, very tiny.
yeah,
It's like
Neat.
it's like all apothecia.
yeah,
That's very interesting.
yeah. Kind of reminds me of Caloplaca flavovirescens, but it's got no thallus. Usually there's a thallus behind the apothecia, and this doesn't have any. So um, a few other things I'm thinking of, but they're deeper orange than yellow. What about like a
This might have been limestone. This one limestone, maybe.
okay? Yeah, yeah. I mean, I can sort of see a little bit of a thallus
So,
underneath it, especially on the one on the back. You can see that distinct pale circular structure. So
yeah.
yeah, I would call it. I guess an iNaturalist is Gyalolechia flavovirescens. Yeah, the first one.
This one here. Okay.
Yeah. Yeah, see, the thallus is much more prominent in this one, but I think that's probably what it is.
Yeah, I was going to say in Lila's, like you can see the thallus in the first one, and then from the other angle, it's more difficult to make out, but
Mm-hmm.
Yeah.
it also looked kind of bumpy, like we were talking about before, rather than areolate.
Hmm.
All right. I'm gonna switch it to that one. Okay. That was a lot. Nice to
Yeah,
have you here,
this is
Jason.
good to do every year. It's every year a little bit more learning happens. Hopefully, you see
Yeah.
that
Yeah.
in all. Jason, I hope you see that in all of us. Like we're trying. We don't have the eye
Yeah.
for it, but
Tiny bit more knowledgeable. It
Yeah.
seems very hard.
Just gotta chip away at it.
Yeah.
I need to re-listen to Jason's talk on lichens that he gave. I need to find that.
Okay.
I think that'll help.
Is
Yeah.
is one of the things that they do is the like the UV test. Is that like mostly on the medulla?
It depends. It is a lot on the foliose lichens. Um, but like for example, Tuckermanopsis, that one's a UV test on the medulla, but Pyxine, that's a UV test on the cortex. So
Hmm,
depends on what the key says.
cool.
All right.
All right. Thanks,
Well,
everyone.
it's 9:09. So thanks everybody who shared, and see you all next week, and have a good night.
You too.
Take care.
Thanks,
Have a good
Jason.
night.
See
Night all.
everybody.